Scientists just found something inside T-Rex fossils that should not exist after 66 million years. Using resonance, Raman spectroscopy with precise 532 nanometer laser wavelengths, researchers detected actual hemoglobin molecules still bound to proteins deep within dinosaur bones. This is not contamination or wishful thinking. It's real organic matter that somehow survived since the Cretaceous period, preserved through goethite mineral formation that created protective shells around protein fragments. But here's what makes this discovery even more significant. The ion-mediated preservation pathway they uncovered confirms and dramatically expands our understanding of what fossils can preserve under specific chemical conditions. The implications go far beyond T-Rex opening new windows into ancient biology that scientists never thought possible. For over a century, paleontologists accepted a fundamental truth about fossils: organic matter disappears completely over geological time. The process of diagenesis systematically breaks down proteins, DNA, and other biological molecules, leaving behind only mineral impressions of what once lived. Bones become stone through replacement processes, and any trace of the original creature's chemistry vanishes into the deep past. This understanding shaped how scientists approached dinosaur research, knowing they could study structure and form, but never the actual biochemistry of these ancient giants. Then researchers began finding something that should not exist. Deep within the bone matrix of Tyrannosaurus, Rex and Brachylophosaurus canadensis specimens preserved in the microscopic spaces between fossilized tissue were molecular signatures that resembled blood proteins. The discovery defied every established principle about preservation limits, suggesting that fragments of 68 million-year-old hemoglobin had somehow survived the relentless march of geological time. These findings triggered intense scientific debate, with many experts dismissing the claims as contamination from modern sources or misidentified minerals. The breakthrough came through resonance Raman spectroscopy, a technique that uses laser light to identify molecular structures without destroying the sample. Scientists direct specific wavelengths of laser energy at microscopic areas within fossil bone, causing molecules to vibrate at characteristic frequencies. Each type of molecule produces a unique spectral signature, creating what amounts to a molecular fingerprint that can distinguish between different chemical compounds with remarkable precision. The key to this analysis lies in the 532 nanometer laser excitation wavelength, which specifically targets the iron-containing heme groups found in hemoglobin molecules. When this green laser light strikes authentic hemoglobin bonds, it produces a distinctive spectral pattern that differs dramatically from contamination or mineral interference. The technique can detect these molecular remnants even when they exist as tiny fragments embedded within the fossil matrix, providing conclusive evidence of their ancient origin. The secret to this extraordinary preservation involves goethite, an iron oxide mineral that forms as hemoglobin degrades over time. As the original protein structure breaks down iron atoms from the heme groups, create protective mineral shells around protein fragments, essentially encasing them in a chemical tomb. This process occurs gradually, with the iron acting as both a catalyst for degradation and a preservative for what remains, creating stable molecular fossils within the surrounding rock matrix. Iron-rich burial environments play a crucial role in this preservation process by creating chemical crosslinks between organic residues and surrounding minerals. These crosslinks stabilize protein fragments against complete decomposition, allowing certain molecular structures to persist far beyond their normal lifespan. The chemistry works because iron forms particularly strong bonds with organic molecules, creating hybrid organic mineral compounds that resist the usual processes of decay and dissolution. This discovery carries important limitations that temper any hopes of genetic resurrection. While hemoglobin molecules can survive through iron-mediated preservation, DNA remains far more fragile and susceptible to complete breakdown over geological time. The double helix structure that carries genetic information cannot withstand the chemical assault of millions of years, even under the most favorable preservation conditions. Hemoglobin's simpler structure and iron content give it advantages that DNA simply lacks. The implications extend far beyond confirming blood protein preservation in ancient fossils. These molecular fossils represent chemical shadows of dinosaur physiology, providing direct evidence of how these creatures functioned at the cellular level. Scientists can now analyze oxygen carrying capacity, metabolic rates, and blood chemistry from actual dinosaur tissues, rather than making inferences from bone structure alone. This chemical time capsule opens entirely new research pathways for understanding dinosaur biology. By examining preserved proteins and their iron mineral complexes, researchers can investigate questions about dinosaur metabolism, activity levels, and physiological adaptations that were previously impossible to address. The molecular fossil record provides a window into the internal workings of these ancient predators, revealing secrets that have been locked away for tens of millions of years. Yet even as scientists unlock these biochemical mysteries, the broader picture of late Cretaceous ecosystems continues to evolve in unexpected ways. The molecular discoveries represent just one piece of a much larger puzzle about late Cretaceous predator communities. The landscape was not the domain of a single tyrannical ruler, but rather a stage where multiple apex predators competed for dominance. While T-Rex has claimed the spotlight for over a century, mounting fossil evidence reveals a far more intricate predator hierarchy than anyone previously imagined. The discovery centers around one of paleontology's most heated debates, the true identity of Nanotyrannus, a smaller tyrannosaur that scientists have argued over for decades. The controversy began with the fundamental question of whether Nanotyrannus represented a juvenile T-Rex or an entirely separate species. Traditional paleontology suggested that these smaller specimens were simply young T-Rex individuals that had not reached full size, but several anatomical features did not align with typical juvenile characteristics. The skull proportions, tooth arrangement, and limb ratios created inconsistencies that made definitive classification nearly impossible using conventional analysis methods. Nanotyrannus possessed larger forelimbs, more teeth, fewer tail vertebrae, and distinct skull nerve patterns compared to T-Rex features that are fixed early in development and incompatible with T-Rex growth patterns. The famous dueling dinosaurs fossil provided the key evidence needed to resolve this scientific mystery. This remarkable specimen, discovered in Montana, contained both a Nanotyrannus and a Triceratops locked in what appeared to be their final battle. The exceptional preservation allowed researchers to conduct detailed analysis of the Tyrannosaur's skull structure, revealing features that would prove decisive in settling the debate once and for all. Advanced cranial nerve pattern analysis exposed distinct neurological pathways that differed significantly from known T-Rex specimens. The optic nerve channels, inner ear structures, and brain cavity proportions showed mature development patterns rather than the expected juvenile characteristics. Growth ring analysis of the bone tissue confirmed that this Nanotyrannus had reached skeletal maturity at approximately 20 years old, when it died with growth rates indicating an adult specimen rather than a developing juvenile. The evidence definitively established Nanotyrannus as its own species. Further research revealed an even more complex situation than initially suspected. Scientists identified not one, but two distinct Nanotyrannus species sharing the late Cretaceous ecosystem. Nanotyrannus lancensis and Nanotyrannus lithaeus each possessed unique cranial features, dental arrangements, and postcranial adaptations that distinguished them from each other and from T-Rex. The name lithaeus references the river Lethe from Greek mythology, acknowledging how this species remained forgotten for decades while being misidentified as juvenile T-Rex specimens. These discoveries meant that 3 different Tyrannosaur species coexisted during the Maastrichtian period, creating a predator-guild system of unprecedented complexity during the final million years before the end Cretaceous extinction. The implications of this Tyrannosaur diversity fundamentally altered our understanding of late Cretaceous ecosystems. Having 3 apex predator species sharing the same geographic range during the same time period requires sophisticated niche partitioning to avoid direct competition for limited resources. Each species likely specialized in different prey sizes, hunting strategies, and territorial requirements to coexist without driving each other to extinction through competitive pressure. Size differences between the 3 Tyrannosaur species suggest distinct ecological roles within the predator hierarchy. T-Rex, as the largest, likely focused on massive herbivores like Triceratops and Edmontosaurus, using sheer power to overwhelm heavily defended prey. The smaller Nanotyrannus species probably targeted more agile, medium-sized herbivores, relying on speed and maneuverability rather than brute force. This specialization allowed each species to exploit different segments of the available prey base without direct competition. The late Cretaceous Tyrannosaur ecosystem emerges as a sophisticated predator guild where specialized hunters filled distinct ecological niches. Rather than a simple monarchy ruled by a single apex predator, this period featured a complex web of predatory relationships that required precise behavioral and ecological adaptations. Yet the success of these apex predators depended on more than just size and strength. Their dominance required sophisticated biological capabilities that scientists are only now beginning to understand. The popular image of T-Rex as a lumbering, dim-witted brute could not be further from the truth. Behind those massive jaws and bone-crushing teeth lay one of nature's most sophisticated sensory systems, a biological masterpiece that transformed this apex predator into a living weapon of extraordinary precision. Recent advances in paleontological research have revealed sensory capabilities that challenge everything we thought we knew about how these ancient giants hunted and survived. Brain endocast studies have revolutionized our understanding of T-Rex intelligence and sensory processing power. Scientists create detailed casts of the brain cavity, allowing them to examine interior skull features without damaging precious fossils. These endocasts reveal that T-Rex possessed sensory processing capabilities that rivaled modern apex predators, with brain structures optimized for hunting and survival in the Cretaceous world. The relative sizes of different brain regions tell a fascinating story about how these creatures allocated their neural resources. The visual system of T-Rex operated like a precision targeting device. Forward-facing eyes provided stereoscopic vision that allowed precise depth perception when attacking prey similar to modern birds of prey like hawks and eagles. The orbital positioning created overlapping fields of view that enabled accurate distance calculation essential for timing devastating bite attacks. Unlike many other dinosaurs with eyes positioned on the sides of their heads, T-Rex evolved a narrow snout that would not obstruct its forward vision, maximizing its visual acuity during hunts. The optic lobes which process visual information were proportionally similar to those found in living birds, suggesting that T-Rex maintained exceptionally high visual acuity. This sophisticated visual processing allowed the predator to track moving prey across varied terrain, detect subtle movements that might indicate hidden animals, and coordinate complex hunting behaviors that required precise timing and spatial awareness. The visual system worked in perfect harmony with other sensory modalities to create a comprehensive awareness of the surrounding environment. Perhaps even more remarkable were the massive olfactory bulbs that gave T-Rex exceptional scent tracking abilities. These enlarged brain structures dedicated to processing smell enabled the detection of both living prey and carcasses across vast distances long before visual contact occurred. The size of these olfactory bulbs suggests sophisticated chemical detection capabilities that would have provided crucial advantages in locating food sources and identifying potential threats or competitors across the landscape. The integration of these sensory systems created a predatory package optimized for late Cretaceous conditions. Stereoscopic vision tracked targets with precision, while massive olfactory capabilities detected chemical signatures from considerable distances. Each system complemented the others, creating redundant sensory pathways that ensured hunting success even when individual senses faced limitations from environmental conditions or prey behavior. T-Rex functioned as a living sensory laboratory equipped with biological tools that represented millions of years of evolutionary refinement. Rather than relying solely on size and strength, these predators succeeded through sensory mastery and precise coordination of multiple detection systems. The sophisticated sensory arsenal reveals T-Rex as an intelligent hunter whose dominance stemmed from neurological advantages encoded in flesh and bone. Yet the true extent of T-Rex intelligence remains one of paleontology's most contentious debates. While brain endocasts reveal impressive sensory processing capabilities, questions persist about whether these anatomical features translated into complex cognitive behaviors or remained within the bounds of advanced reptilian intelligence. The answer may fundamentally reshape our understanding of how these apex predators actually lived and interacted with their world. Evidence from trackway discoveries and bone bed analyses suggests these apex predators engaged in more complex social behaviors than the solitary hunter stereotype implies. Fossil sites in Montana and Alberta have yielded multiple T-Rex specimens found in close proximity with bone assemblages that indicate group activities rather than random individual deaths. These discoveries challenge the fundamental assumption that tyrannosaurs were purely antisocial creatures driven by territorial aggression. The debate over T-Rex intelligence has become one of the most contentious topics in modern paleontology. Early neuron count estimates initially suggested cognitive abilities that might support complex social structures, but these calculations have since been challenged as significantly overinflated. Research teams led by scientists like Kai Kasper have provided corrected estimates that place T-Rex intelligence firmly within reptilian parameters rather than approaching mammalian levels. Recent studies align T-Rex brain capacity more closely with modern crocodilians than with primates or other mammals. The revised calculations account for fundamental differences between reptilian and mammalian brain architecture, recognizing that neuron density and processing efficiency vary significantly between these vertebrate groups. Crocodilian intelligence, while sophisticated within reptilian parameters, operates through different mechanisms than mammalian cognition, focusing on environmental awareness, territorial behavior, and opportunistic hunting strategies rather than abstract reasoning abilities. Some evidence suggests possible coordinated behaviors, though the extent of these social structures remains speculative. Bite mark patterns on herbivore fossils and the distribution of Tyrannosaur remains across specific geographic areas hint at territorial associations, but definitive proof of pack hunting strategies remains elusive. The survival of some T-Rex individuals through severe injuries that would have prevented normal hunting activities raises questions about potential group assistance, though such behaviors could also result from scavenging opportunities rather than active caregiving. The discovery of multiple Tyrannosaur species coexisting during the Maastrichtian period adds complexity to understanding social dynamics and interspecies competition. With Nanotyrannus lancensis and N. lithius sharing territories with T-Rex, competitive interactions between these species likely influenced territorial behaviors and resource allocation. However, these relationships probably operated through instinctual responses and learned behaviors rather than conscious strategic planning. The balance between advanced sensory capabilities and reptilian cognitive limitations creates a nuanced picture of T-Rex behavior. While these predators possess sophisticated sensory processing systems that enable complex environmental awareness, their brain architecture imposed constraints on abstract reasoning and long-term planning abilities. Social behaviors likely developed through instinctual responses and experiential learning mechanisms rather than deliberate decision-making processes comparable to mammalian intelligence. T-Rex emerges as what researchers describe as reptile smart, possessing behavioral complexity comparable to intelligent crocodilians and monitor lizards rather than primates. This intelligence enabled sophisticated environmental awareness, territorial recognition, and possibly some forms of social interaction, but within the evolutionary constraints of reptilian neural architecture. The predators likely developed survival strategies that maximize their advantages through instinctual and learned behaviors rather than abstract reasoning. This understanding presents T-Rex as a behaviorally sophisticated predator operating within reptilian cognitive parameters capable of complex environmental responses without requiring mammalian-level intelligence. The evidence suggests these ancient giants possessed remarkable adaptations that made them supremely effective predators, challenging our preconceptions about prehistoric life while remaining grounded in scientific reality. As researchers continue examining these discoveries, the accumulated evidence reveals creatures far more complex than simple movie monsters. These scientific revelations fundamentally reshape our understanding of late Cretaceous predators. Molecular preservation techniques reveal blood proteins locked in ancient bone, while taxonomic revisions expose a complex Tyrannosaur dynasty rather than a single apex ruler. Behavioral analysis demonstrates effective sensory adaptations and potential social structures that operated within reptilian intelligence parameters. These discoveries collectively paint a portrait of biological sophistication that emerged through millions of years of evolutionary refinement. Advanced analytical techniques continue to extract secrets from fossils once considered scientifically exhausted, suggesting that our understanding of Cretaceous ecosystems remains in its infancy. The age of dinosaurs was far more intricate than anyone previously imagined.